Okay, so Dr. Zheng is one of the professors at the GCCRI. He does a lot of really interesting work with fusions and fusion genes, and that's a huge deal for pediatric cancer research. And that's where in the GCCRI we mostly do a lot of pediatric cancer research because a lot of them are driven by fusion genes. So he's going to be talking a little bit about how he uses RNA-seq to do that and some other aspects of alignment that are really interesting and really advanced. You're not going to actually have to know how to do this stuff, but I think it's really exciting to see what you can do beyond just simple gene expression like we've shown you so far. So with that being said, Dr. Zheng, take it away.
Okay, so since almost half of the audience is from my lab, and if I plus you guys, then most of the people I actually already know. All right, so anyway, I think this is going to be really easy for me.
We make sure we give you some hard time, so your life.
So, really, I think what I want to do really is that, I wanted to make it an open discussion about what we can do with gene expression data, because this seems to be the most popular platform around here, and plus if you think about the single cells, I think discussing the potential of RNA sequencing can be quite interesting if you think how you can transfer these ideas to single cells. Right?
Yeah, I do. I mean, echo you. I think the most difficult thing is after you've done the differential expression analysis, then moving to the beyond that, just simple looking at which gene is up and down. I mean, some pathway analysis. It's like when you're going to end your analysis. So, but that's true. So it should be most interesting.
So for those who are not coming for this lecture, I think they're going to regret it.
Absolutely.
Okay. All right. So I think what this series has covered so far is our basic R and data visualizations, and you have learned a lot about RNA sequencing, including the data structure, some basic concepts of alignment, expression quantifications, differential expression, and so on and so forth. You also learned something about the Linux shell commands. But if you talk about what you can do beyond gene expression, I think there are really two aspects to this question. So first is, for the RNA-seq per se, what other information you can get other than gene expression from RNA-seq? That's the first question. First aspect of that question. The second is, once you have the gene expression, what can you do with the gene expression? I think, actually the first aspect is quite straightforward. We will talk about what else you can get from the RNA sequencing. But for what else you can do with gene expression, I think it's more about the integrative part of data analysis. So because what this course has covered is gene expression and differential expression. But if you only think about gene expression, then the actual first high throughput technology that give you gene expression measures is microarray. And microarray was almost that was invented more than 20 years ago, 30 years ago, I guess. So I still want to bring this up because for the microarrays, if you only need expression, then you can pretty much use microarray. And people, when the RNA-seq first emerged, people just keep saying that the RNA-seq is revolutionary and is so much better than microarray. But when you look at the gene expression, actually, I don't think the RNA-seq brings so much more than microarray, other than it has a bigger dynamic range. That's I think many of these techniques we use nowadays for RNA-seq actually come from microarray. But the way RNA-seq and microarray give you expression data is the microarray uses fluorescent signals, and RNA-seq give you the sequence, the short reads. And that's the key difference between the two platforms, because what else can RNA-seq offer actually come from this short reads. Now, what the short reads have is ATCG, of course. And also the reads are derived from the cDNA segments. Giving this read counts, I mean, there are two things, right? Read counts and this nucleotide sequence. Given the read count and the nucleotide sequence, what you can get is, at least you can get gene expression. And if you combine with the nucleotide, you can get allele-specific expression. And also you can compare the sequence from your RNA-seq with the reference genome, and so that you can get this variance. Now this variance can be mutations, RNA editings, and if you have a control, then you can get, if you don't have a control, then you can get the SNPs, the expressed SNPs. But if you do have the controls, then you can have a sense of whether this variant is a mutation or it's RNA editing, should you happen to have the DNA sequencing data. Now the aberrations in RNA or in cDNA will be carried over to this reads. Therefore, from this reads, you can infer the occurrence of gene fusions and alternative splicings. So I'm gonna really go really quick because I think most of the stuff you already know. Now the factors affecting RNA sequencing performance is, of course, number one is the total number of reads you get from the sequencing. And the read length also is very important. So if your goal is to discover the gene fusions, and longer reads are much better and powerful than short reads. But if your goal is to estimate expression, then the read length doesn't really carry too much weight in that. And another is whether the RNA sequencing is pair-end or single-end. Now, pair-end is apparently more powerful and more accurate. And one aspect that I think people often ignore is whether you have the reference genome or whether you don't have the reference genome. Now, when I say reference genome, I know most of us work with human cells. What I really mean is whether you have an accurate file annotations for the gene models. How accurate are the exon boundaries, the UTRs, and so forth. Now, assuming technical glitch is not an issue in those experiments. So I'm going to skip this because I think how the sequencing data look like has been shown by other lectures. What I do want to say in the next two slides is how the alignment looks like. So this, at the bottom of this slide, this is the gene model in the reference genome. So a typical gene has 5' UTR and 3' UTR. And these are UTRs that do not translate into proteins. That's why they're called UTRs. And this is the transcription start site. And anything before this transcription start site are UTRs. And anything after the stop codon is 3' UTR. In between, you have this introns and exons. And these introns and exons are the template that translate into proteins. Between the exons are introns. Now in alignment, you actually get the short reads aligned to each exon. Okay, but this is what the gene looks like in the genome before splicing. Now, I want to say the transcription start site of 5' UTR is not necessarily in exon 1. It could be in exon 2 or exon 3, depending on the gene. In some cases, the first 2 or 3 exons can be all in the 5' UTR region. So therefore, they're not translated. So if you look a little bit deeper into the alignment, this is what we have shown in the previous slide, and the reads are aligned to exons, and some reads are aligned to exon boundaries, part in the intron, part in exon. But this is what the gene looks like in the reference genome. But in reality, what the gene, before you sequence the sample, what the gene look like is actually here, like this. So the sequencing protocol captures the mature mRNA, then first reverse transcribe them into cDNA, because cDNA is more stable than mRNA. And what you sequence is actually those cDNA molecules. Now, when you have the cDNA, the typical RNA sequencing protocol has a step that fragment the cDNA molecule into this random pieces. So these fragments are the input material for your sequencing. And the reads actually are derived from such fragments. And in our realignment, basically, we put this reads back to the segments. And each read reminds you of the segment it is derived from. So if, assuming the RNA is perfect and there's no aberrations, and what you would expect is that the reads are all aligned to exons or junctions, but not introns. This is not, right? So, but in RNAseq, it's pretty common. You see the reads are aligned to introns, and part of the reason is this alignment can have some noises, and you know, the aligner sometimes assigns this reads to this intronic regions. It's also possible that you have this unannotated exon, and this exon is transcribed, and then the RNA sequencing capture those exons. So, and we expect all the read pairs aligned to the same transcript, the same gene, the same transcript. But if your alignment tells you that this pair end reads come from two different genes, and that's the indication of a gene fusion, because typically they should not come from two different genes. And they should follow an expected insert size, because every RNA sequencing, they select for a typical insert size, which is around 300 to 500 base pairs. So if the alignment, if the reads actually are aligned to very distant regions, that will be indicating of either read-through events or alternative splicings. And if the reads do not follow the expected orientation, and that's also an indication of structural rearrangements, in this case, gene fusions. Of course, when you see the nucleotide from the short reads do not match those of the reference genome, that indicates either a somatic mutation or editing, or a variance, the SNP. But I would say differentiating a mutation from editing is not that easy, because you do need the DNA level data to ensure this variant is not happening on the DNA level. Rather, it's only happening on the RNA level. That's called RNA editing. So if you look at this short reads, now, assuming we're doing this pair end resequencing, you really have to imagine this as a two-strand of DNA. One is from 5' to 3' direction, and the other is again from 5' to 3' direction. So in this diagram, the blue represents the forward read, which is the minus strand basically. The red indicates this is minus strand. Sorry. The blue is the plus strand, and this red is the minus strand. So the plus strand is also called forward strand, and the minus is also called a reverse strand. Now, I think this is a little confusing. I don't know if you learned this in molecular biology class, but the forward strand and reverse strand is not equivalent to sense or antisense strand. Because when you talk about sense or antisense strand, that is relative to where the gene is located. Okay, so if the gene is located on the minus strand, then the R strand is basically your sense strand. Now this is very important because for the identification of gene fusions, you're not only identifying the fusion, the two genes that are involved in the fusion, but also you need to determine which gene come as the 5' gene or which gene come as the 3' gene. So basically, you need also to determine the direction of the fusion event. So if RNA sequencing is so powerful, then why would we just do RNA sequencing instead of doing, DNA sequencing? Should RNA sequencing be like our best platform? And it turns out that RNA sequencing does have limitations. And this is, a very brief step-by-step procedure for RNA sequencing experiments. I list on the right some of the caveats in the RNA sequencing protocol. For instance, even the fragmentation sometimes is not uniformly random. So you would sometimes have this bias towards some site, because the fragmentation is, if you have, for instance, in the genome, you have this fragile site in the genome. Now, data has shown that for this fragile site in the genome, they easily break. On the DNA level, you would expect a lot more breakpoint around this region. This creates many copy number events. And for the RNA, this is pretty much the same, because as you have DNA level events, that can be transcribed into the RNA level events. Do we have, okay, right. Okay, so, and of course, PCR efficiency may also be different for different transcripts. And especially in the cDNA reverse transcription step, RNAseq can introduce many artifacts that in the final RNAseq data would appear to be variants. So now the gene fusions, I think, I studied gene fusions since 2011. And so the gene fusion is really in the cancer genome is really a remarkable phenomenon, because it connects to different genes and creates a new transcript. Now, ever since the introduction of RNA sequencing, looking for new gene fusions has been a major question for the field. And the question is, can you design a better tool to identify gene fusions? But in general, I mean, if you talk about gene fusions, it's pretty common in human cancer, because you can consider gene fusions as a result of genomic instability. But if you talk about the driver gene fusions, then the adult cancers do not have that many driver gene fusions. Typically, the fusion events are less frequent. There's quite rare to see recurrent gene fusions. Now, the gene fusions, especially the driver gene fusions, are more common in children cancer than adult cancer. But for some specific subtypes of adult cancer, the fusions can be very frequent as well. For instance, high-stage prostate cancer has a fusion called TMPRSS-ERG. That's a very common fusion, and roughly 30% of the high-grade prostate cancer has this fusion. Fusion is important also because one of the major success in precision oncology is the very first molecular targets in target therapy is a gene fusion, targeting the gene fusion. That's the Philadelphia chromosome BCR-ABL fusion. And it turns out that when you target this fusion, the disease remission is quite durable because fusions are usually very early event, very clonal. So when you target cells that carry these fusions, you're targeting the initiating cells. I would say in adult cancer, looking for these targetable fusions have been largely unsuccessful, because the example I give, for instance, TMPRSS-ERG, that's a very frequent fusion in prostate cancer, but you can't really do much to this fusion because the fusion involves a transcription factor, and if you target the transcription factor, the cell dies. There are just too many side effects to the targeting. The same is for Siyuan sarcoma. EWS-FLI1 drives the Siyuan sarcoma. Ewing sarcoma is a very rare type of childhood cancer, and more than 90% of Ewing sarcoma tumors have EWS1. But again, that's a transcription factor fusion that nobody can really do much to this fusion, at least at this current stage. Now, how do you identify gene fusions? Remember, I said in the previous slide, the abnormalities you see in the RNA sequencing, some of those are actually indication of gene fusions. So you can apply this rationale into the fusion identification. The first evidence of gene fusion comes from this we call discordant read pairs. So if you have two genes and you have a pair of reads, and each read from this pair comes from or aligned to different genes, then this would indicate that the gene A and gene B potentially somehow form one transcript in your sample. And that is an indication of a gene fusion between gene A and B. Now, if you only rely on this evidence, that wouldn't be enough, because misalignment is pretty common. It doesn't have a huge impact on expression level estimate, but if you rely on discordant read pairs, and for every tumor sample, I'm pretty sure you're going to get thousands of such fusion candidates. Another evidence comes from the splicing reads. It's junction spanning reads. So assuming you have two genes, and you can, if you suspect there is a fusion going on between these two genes, now what you can do is you can construct a library consisting of all the exon boundaries between A and B. So you can build basically a small reference genome that contains all these possible exon boundaries, and then you align your reads to this library of exon boundaries. Now, if you do find reads that span this library, this exon boundaries between these two genes, and that would significantly increase the confidence level of a fusion between those two genes. So we typically rely on these two pieces of information to tell whether there is a fusion between gene A and gene B. But the issue with gene fusion is not to find the proper candidate. You always find the proper candidate. The issue with gene fusion is almost always that how do you filter out these potential artifacts? So typically, in a cancer genome, you actually get thousands of potential candidates. And if you apply this very conservative or strict criteria, filtering criteria, so you typically end up with only maybe a dozen of candidates, meaning the vast majority of these candidates are not real. So how do you filter out these artifacts? That's a big challenge for the fusion identification. So actually, I jumped a little bit. So there are many tools to identify gene fusion or call gene fusions from the RNA sequencing data. Our tool is called Prada. STAR has a tool called STAR Fusion, and Fusion Catch is also quite popular. Diffuse is one of the earliest tools to call fusions, and relatively young tools include ERIC, Script, and Java. Top Hat Fusion is quite old as well. And of course, this relies on the top hat alignment. Now, one thing when identifying the fusions, typically fusion identification relies on alignment. So for any tool to perform properly for fusion calling, it has to start from alignment. So even you have the BAM file, I won't trust my BAM files and directly feed my BAM files into the fusion calling tools. And most of the tools they don't really need your BAM files. Even you give them BAM files, they actually convert them back to FASTQ, then redo the alignment and then calling. Okay, so that's what we do in the product. We don't really rely on the BAM files provided by the user. So like I said, one of the challenge with fusion is you have to remove these possible artifacts. Now, the criteria to use to remove artifacts, there are many. For instance, we typically consider, if a gene is found fused with too many partner genes, and this typically indicates the fusion is due to some misalignment or due to the sequence of this gene being, too homogeneous. And tumor normal, if you have the normal samples, that's also a good good idea to remove fusions you see in both tumor and normal samples. Imagine if you have some issue with one gene's sequence in terms of fusion calling, then you would expect the same bias would be existing in both tumor and normal. In that case, having this normal samples would be a good idea to remove such artifacts. And also, I think another consideration is the fusions can be the product of a copy number polymorphism. So in that case, the fusion can be considered a germline event. So GPR98, that's a very typical germline fusion event that is usually seen in both normal and tumor samples. Another issue is highly expressed genes. So why the expression level of the gene has anything to do with gene fusion? The reason is, with highly expressed genes, you have a lot more reads for that gene. Now, when you have more reads, it's more than likely some of the reads come from misalignment. And the more reads you have, the more likely you have such inaccurate alignment. So in that case, if you see a highly expressed gene, and only with few reads from this gene indicating there is a fusion involving this gene, this typically indicates this is artifact. Of course, if you have two partner genes with very similar sequences, especially genes from the same gene families, and these are typically due to the similar sequences between the two genes. Identical junctions, this is not entirely impossible in the cancer genome. Just like hotspot mutations, there could be hotspot fusions. But for a gene, for fusion, if you see, let's say you identify a highly recurrent gene fusion and you're excited, this might be your big finding. Now look at the junction. If the junction is all identical among these events, it's more than likely this is an artifact. Remember, the fusion is very similar in a way to DNA rearrangement. It is a random process. So if you see the same identical junction in the fusion candidates, this, from a statistical point of view, this is very highly unlikely. Again, too many fusion partner genes, one of our last resort is, let's say you identify these fusions candidates and you have designed this filtering criteria to remove these potential artifacts. Even you apply them, you can accidentally remove the real fusions, for instance, and you can also fail to remove the known artifacts. What do you do then? We also, the last resort is we create a positive and negative fusion list. So the positive fusion list are the known fusions that by no means should be removed from your fusion list. The negative list is include the fusions that we know they are not real. So even if they survive all your filters and we remove such fusions from our results. So this is really the last thing we use, called positive and negative fusion list. In the new version of product, we actually include this. So the positive fusions come from the literature. We more or less have about 300 positive fusions, and the negative fusions we have, also a couple hundred negative fusions, I believe. So if you this by no means all I said before have included everything possible you can get from RNA sequencing. I didn't mention how you can get allele specific expression, or how you can do the QTL, EQTL expression analysis. But I think one of the power of RNA sequencing analysis, if you go beyond gene expression, is how you can integrate the expression data with other type of data. Now you can consider expression as the functional readout of genetic and epigenetic change. Now I think you can argue that if you can read this, you can analyze this functional readout of this genetic and epigenetic events, and why not just go after this genetic and epigenetic events directly? So that's the approach taken by, for instance, TCGA, because they rely heavily on DNA sequencing and copy number change. But I think more and more people realize the expression is very complicated, but it actually gives you some information that you can get from DNA and from DNA data. The expression, of course, bridges genetic and proteomics data. You can integrate the gene expression with other type of data, including the survival and other things. So I think that's pretty much all I want to talk about. Okay, so now the next part, what I want to do is actually spend quite some time on this. Is I want to give a demo of how to visualize gene expressions in IGV. So I know we have analyzed all the TCGA data, so we know exactly which TCGA case has which fusions. So that should not be an issue. So I, with Dr. Cheng's help, I downloaded some BAM files from GDC. And I don't need to download all of those BAM files. I only need a small slice of those BAM files covering the gene regions of the regions of the fusion genes. But what I found was,
would you mind actually showing how you got those? How you did that on GDC?
Yeah. Okay. That's good. Let me see. Okay. Okay.
But before you actually leave the topic, I have a few questions, sort of a back on the name. So I just wondering if you can comment on the that there are like fusion panels, specific design to those are mostly. I only know Illumina, but I haven't looked at the other panel. Mostly I capture only looking at the known junction point, right?
Yeah.
Or they have some new things, or the
no, that's the interesting thing. So if you wanted to use DNA panel to capture this RNA fusions, you have to capture you have to cover all the intron regions between this known between the two exons, because you don't know where the breakpoint is.
Yeah.
It can be anywhere in the intron. So that's why you need to design this region to specifically include this region to be able to call fusions from the panel. Yeah, that's why most of the panels, they are not able to capture fusions.
They are not looking at the RNA, or they are looking at. I thought they are.
Yeah, if they can do RNA sequencing, that's a lot easier. But for some panels, for example, the MSKCC, Memorial Kettering, has the impact panel. That impact panel can detect a few pre-selected fusions, and they have to include these this intronic regions to be able to do that. But most of the panels from most of those cancer gene panels don't do that. Yeah.
So the other kind of quick question as well. So so if you looking at the so now the long read sequencing gradually getting in there, even though the accuracy is still kind of problematic. But for fusion, wasn't that? I mean, that sounds like it's ideal. Then just no way.
No doubt, the long read sequencing would be ideal for sequencing and also for alternative splicing analysis.
Alternative splicing.
Yeah, I think I know for sure. I know people are doing it. For example, the Jackson lab, they have a genomic development center or something like that. In Connecticut, they have a new campus there, which was set up a few years ago.
Yeah.
They are doing this long-read sequencing to understand how splicing works. So if the long-read sequencing becomes mature enough or cheap enough, I think the splicing will be our understanding of splicing will be on a different level.
Yeah. So
because
that's
right now it's only rely on the it's a very limited ability to do that.
Yeah. Haven't you? So you didn't see any kind of a data resource that kind of, let's say, taking a panel of the cell line was probably known, just go through it. They didn't. Nobody have done that. Oh, okay.
No. One thing I think, do you know there is a consortium called T2T consortium?
T2T. Okay.
This is called telomere to telomere consortium.
Oh.
They're not doing this long-read sequencing for RNA. They're doing this for DNA. What they want to do is basically to construct a complete reference genome for each chromosome from one telomere to another telomere. It's called T2T. So if that's complete, then estimating telomere lengths, or that will be just. It's not even an initial, and it's not even a thing. You basically just count it. That's it.
Just count it.
So,
yeah.
But the T2T, I understand from what I from what I recall, the T2T consortium now is only working on one cell line, I think. So I think I don't really know much about it, but I think it involves a tremendous technical challenge to assemble, T2T for the entire genome, even for that cell line.
Long read sequencer still it's still having some difficulty.
There's a long way to go for the long read sequencing. And the PaxiBio, they have been running their platform for quite some time now, right? But it's still it's good for microbiome genomes. It's still not good for humans because you get a much longer read for microbiomes than the humans. The humans average, you get several K, the average read length. But for the microbiome, that would be really good. And you have millions of base pairs from the microbiome, but you only get like 20K or 30K by average for the human genes. So I don't know what is the bottleneck of that, but that's. I think at least 2-3 years ago that was the status quo for PaxiBio.
Yeah, I was thinking if that's the way someone would start accumulating those resources, if that would sort of create a standard, at least go through the cancer cell line rather than the expression and just say we got all the variant that you need to get it. Okay, good. I mean. Yeah, just move on. I just got a few questions.
Okay, so Henry, I think Henry has a very good question about where you can download the spam files, or where do you get the slice of the spam files. So this is GDC, but let me start from beginning. If you, I think I recall, I recall Henry mentioned GDC in one of the lectures. GDC full name is called Genomic Data Commons. So you can just Google NIH GDC. So this will bring you to this data portal called Genomic Data Commons. Now, this is officially sponsored by NCI, so the data is heavily biased towards cancer sequencing data. So what you can do with this data is, let's say I wanted to, download the TCGA data. Then after you click TCGA, then you see all the disease types that was included in TCGA. Then you can click any, and this will bring you to a summary page of all the data that were generated by the TCGA consortium for this disease. Let's say I am interested to download a few BAM files. So what you do is you see under this data summary page, you have this different type of data, like a sequencing reads, copy numbers and DNA methylations and so on and so forth. The data also is organized by the experimental strategy. So whole genome, whole exome sequencing, and so if you click, for example, if you click RNAseq, let's say I want to download one BAM file from the RNAseq, you can click here. You can do many different ways, and it will come to the same page essentially. So let's say I'm just go directly for some BAM files, some RNAseq BAM files. Basically, what you do is click here, and you can see this. All these files are related to RNA sequencing, and you can narrow your search by, let's say, I'm interested in BAM files. So if you click BAM files, and that's all the BAM files for the TCGA ovarian cancer RNA sequencing data. Now you can see on the right, these are all the files you can download, but you can see there is a column called access, and these are controlled access, meaning you need credentials to download this data. If you have credential, then you can log in here, and then this will essentially become accessible to you. Now, unfortunately, my credential expired, so I can't show you how I can download this. But if you click here, so you can see these are the metadata for that particular sample. Okay, so even though I don't have credentials to download the BAM files, I can download this expression data for this case. Okay, so if you don't, I don't think it's okay. Right here, if I do have the permission to download the data, you can download the whole BAM. But if you don't need the whole BAM, then you can download a slice of the BAM. This is called the BAM slicing right here. Okay, so what the BAM slicing does is you give a region to BAM slice, like here. Let's say I'm only interested in this region. Say this region covers my favorite gene EGFR, then I can just put this region in here, and then I can just download, and it will download it. Okay, so this is how you can download a slice of the BAM file. Oh, it turns out I can. Yeah, I can. Okay, all right, that's good. Now, this of course involves a lot of manual clicking. You have to click this and click that. And imagine if you have to download, 500 cases, and that would be not a viable option to you. So the GDC provides a tool called GDC Tools. I don't think I can show it here, because to use the tool you need a Linux. You need to install the tool on Linux. And then you have to provide a list of files you want to download using this UUID. This is a unique identifier for each file. So they have the UUID for each case and even each file. So once you get a list of these UUIDs, and you can put all them in one file and then give it to the GDC tools, and then GDC tools can download all that for you. Let me see. In the example, so in the folder, I included I have a download bash file. This is essentially if you have access to virtual machine, or you can even do this on your own Mac. And the curl is basically a generic tool, wrapped in every Linux distribution. So you can download the slice of BAM here. Now, if you have the token file is basically the credential file that tells the GDC you have the permit to download such files. You give this token file, you type this token file into the command line, and then you can run this command to get your slice of BAM. Okay, so now getting back to the demo. So I told you I was trying to download the BAM files to show you how the fusion reads look like in IGV, but it wasn't very successful because I realized that even for the cases where I know the fusions exist, after downloading the BAM files that cover the both gene regions of the fusion, I don't see any fusion reads in that small BAM. It turns out, then I went back to, our BAM. So we analyzed 1,000 target cases, and all the BAM files are on our own server. So I went to the BAM files, and I got the slice of BAMs from our own BAM files, and still there's no fusion reads in those BAM files. So I was very much baffled, and so I went back to star. All these are, use star for the alignment. Now I think I realized that the star, unless you tell star specifically to write this reads into the BAM, it won't write this fusion reads into the BAM. It writes the fusion reads into a junction file called junction. The information is included there. So that's the reason, even when I download this BAMs, I won't be able to see any fusion reads in my BAMs. But let me show you something else. So luckily, I have some BAM files from my previous analysis, and this BAMs were generated by a different tool. So all the reads are here, but what I can show you is only half of the fusion reads, because I have this BAMs only for one specific gene, not for every, not for both genes. But let me show you. Let me show you how to visualize it. So one thing I want to remind the class is, remember I said to use the reads to identify the structural rearrangements, a particularly important piece of information is the read orientation. This will be reflected in the IGV, but let me show you how the IGV looks like. Where's my IGV? Oh, okay. So this is my IGV. I don't know, Henry, is there a way to share the entire desktop so that I don't have to?
Right, you should be able to share the whole screen instead of sharing specific windows.
So let me see.
So it should be like screen 1, screen 2, screen 3. It should say something like that. It should be like one of the very first options when you hit share screen.
Oh, yeah. Okay, right.
Awesome.
So this is the screen, right?
Yes.
Yeah. Okay. So see, these are this is my IGV. For my BAMs, it's from HG19. So I'm going to switch to HG19 from IGV. And these are the small BAM files that have been uploaded to the box folder by Henry. So I'm going to load one of those BAMs. Now, to use IGV to visualize BAMs, you need not only the BAM file, but also the index file. So the BAI indicates this is an index file. So you load this into IGV. And now, because this is a small BAM, the reads are not, not available for every gene, but I know where the reads are. So I'm just gonna go directly to the read. So here you can see these are the reads aligned to TERT gene, this one gene. Now all these BAMs only have reads aligned to TERT. Now you can see that's why I only can show you half of the fusion reads, and I know in these cases, TERT gene is fused with another gene, but I don't have reads for the other gene, but I can only show you the reads for TERT. This is a very interesting gene. I wouldn't go over all the biological significance of this gene. But when you look at the reads aligned to the TERT gene, what you can see is pretty remarkable is that the gene is on the minus strand. Okay, so the direction goes from the right to the left. Now, normally, what you would, even though for the regular RNA sequencing, you have more reads aligned to the 3', but for this case, the first half of this gene is remarkably clean. Now, why is the case? Because the first half of the gene is fused out in the fusion event. Basically, this half is out, so it's not expressed. But in the read, if you can color this read, for instance, let me zoom in just a little bit. So every every rectangle indicates this is a read, and you can see this directions. Direction indicates whether the read is aligned to the forward strand or is aligned to the reverse strand. See, this arrow pointing to the left. These are the reads aligned to the reverse strand, whereas this read with their arrow pointing to the right, indicating this read is aligned to the forward strand. Now, when you can color the alignment by insert size and pair orientation. So this is very, very important because with sequencing, if you want to use sequencing to identify structural events, the insert size and the pair orientation is a very critical piece of information that allows you to identify such events. So when you color them, you can see my reads, they're actually colored differently. This is one group, and this is one group, and this is another group. And if I zoom out, you can see on the left, IGV shows you this is RR, this is LL, okay? And this, without LL or RR, this indicates these are the normal reads, okay? So at least based on the read orientation and insert size, IGV does not think these reads are abnormal. But the RR and LL, they are the abnormal reads that was highlighted by IGV. So what you can do is, in IGV, you can visualize the matte region in the split screen, which means it can bring you to where the other end of, the other read in that pair aligns. Now imagine if for a pair of reads, one aligned to this gene and the other aligns to the other gene, then this will basically tell you where the other gene is or which gene it is. So, if I do click this, then you can see, and for this pair of read, one is aligned to TERT and the other is aligned to a gene called SLC30A5. Now, I said I don't have reads for this region, that's why it looks so clean, but we know in this sample, there is a fusion event between TERT and SLC30A5. That's how you visualize the fusion event in IGV, okay? That's pretty much it. That's really simple. I mean, once you know where to click, that's really simple. Now you can do a lot of things. I don't know if Henry, if the class has actually has some tutorials on IGV, and I think this is a really, really useful tool to visualize not only the sequencing data, but also the copy number data and gene expression data. So I really enjoy using IGV for my copy number analysis. Actually, I can do a great deal within IGV without writing any code for copy number analysis. But anyway, this is how IGV looks like, and you can see here all the reads. If you let's zoom out a little bit, so you can see at the bottom of this interface, you have this gene models. Okay, so each box indicates this is an exon. Okay, this is precisely how the TERT looks like in the reference genome, and you can see most of the reads are aligned to the exonic regions in TERT, and you don't see, even though sometimes you see spontaneous alignment of these reads to this intronic regions, but if you look at the overall coverage of this RNA-seq, then you can see the major coverage is seen in this on top of this exons, but not in this intronic regions. Now this intronic regions pretty much looks like noises from the alignment. So I highly recommend the group to use IGV because I think it's visualization helps a lot with finding the right patterns to go further beyond the expression. Okay, So that's pretty much it. I don't think we have time to go over this website I have listed, but the website the websites I listed are pretty easy to use, and you can integrate, like I said, you can integrate a lot of things with gene expression. So maybe just a very quick overview. So this is a website we developed for gene expression analysis. So for instance, you can click here. I mean, it's very intuitive. What you can do is you basically correlate gene expression with all sorts of things. So for instance, you can you can look at the one gene's expression across these different disease types. In this case, if you look at the PTGFRA, then you can see the PTGFRA expression is high in extracranial rhabdoid tumor. That makes a lot of sense because the PTGFRA is a marker of mesenchymal cells. So if you switch, you can switch data set here. The PVTC previously I showed was a collection of models, and this is the patient data, and you can convert the data to log scale, and you can order them differently, and it can do all sorts of things. And the data, of course, allow you to download the PDF, download the actual data behind this figure, and so forth. So you can do a lot of things with this, and you can even do survival analysis, correlating your gene expression with survival, and you can do multivariable analysis, adding the covariate in this box, co-expression, and correlate expression with mutations and copy number. So in a way, I think, partially addressing Dr. Chen's question in the beginning, once you have the gene expression, what else you can do? I mean, if you have the gene expression and only phenotype, I think pretty much all you can do is, find the differentially expressed genes or do the pathway analysis. That's pretty much all you can do. I think the integration, the power of integration depends on how much data you have or how many types of data you have. The more data types you have, the more you can do with integration. And of course, the hypothesis is also important. You have to know what you're testing before you actually do it. So that's pretty much it. I was intending to show you the R2, I think, which is very similar to our portal, but the R2 is has more data, but the interface is ugly. But anyway, and there are other tools. I think the GCCRI offers a well, I guess I'm not this year, maybe next year. We will offer a seminar or lecture on using this GUI tools, the websites basically, to do data analysis or tell you how to find this data. And if you're interested, just sign up. We will send out information and notification once we figure out where to host this seminar, a lecture, and so on. So I meant to have an open discussion about the gene expression analysis today, and you have my emails here. So if you have questions, you can write me emails, or you just schedule time to discuss any issue, and I'm sure I can give some comments on the data analysis. So let me know. That's pretty much it. So I give this to Henry, because I know you have some announcement, right?
Sure. Well, this is great. I wanted to see, just because we covered a lot, I wanted to see if any of the students have any questions or had any issues getting IGV to work with those BAM files. Okay. Does anybody have any questions about why you would use IGV, or like kind of what the purpose of doing this genomic, visualizing it at this genomic level would be? I was interested to see there's one thing you can do with IGV that I thought would be kind of interesting to show, which is this sashimi plot.
Yeah. Oh, yeah. That's a great point. Let me show the sashimi plot.
Okay, great.
Okay, right. So let me load my IGV, and so
Anyone knows why they call sashimi?
yeah, because it looks like sashimi, and sashimi is a Japanese cuisine, right? So let me see which BAM is slightly bigger. Okay, this is
Yeah, and I meant to ask you, what's the other file? I know one is showing clear fusion, but the other one looks no fusion, right?
No. That's what I'm saying is this file, this BAM files, I call this a mini BAM because I generated this files, 3 years ago.
Oh.
I only kept the third gene. I didn't keep the other gene. Because the reason I kept this files, I kept the third gene because I was interested in studying the alternative splicing of TERT, only TERT. So that's why I only kept the information on TERT, but not any other gene in the genome.
So the other have the now it's a new one. You have the other gene information there. Okay.
This one is all the BAMs I have is on TERT. So yeah. So okay, this is the RNA alignment on the TERT gene, and what you can do is you can if you right click, and there is an option called sashimi plot. So when you click the sashimi plot, IGV will generate this plot for you. So at the bottom is the gene model for the gene you study. So you can see this. There are two isoforms for this gene based on the annotation IGV uses. Now the difference between the two isoforms is this one, this exon. There's one exon here in the second isoform, but there's no such exon in the first isoform. And on top of this figure shows you how many reads are connecting different exons, okay? For instance, here you see you have 18 reads connecting the first and the second exon. Now, if every exon junction is, if you don't have any splicing event, then what you would expect is all the junction spanning reads or the exon joining reads would be jumped from one exon to the next exon, right? So from here to here, from here to here, and then go on and on. But if you do have alternative splicing, then you see these abnormal junction spanning reads, like you have reads connecting this exon to this exon. If there's no alternative splicing, you wouldn't expect to see such connections. But because of alternative splicing, there must be a transcript or isoform that connects this exon directly with this exon without having these two exons. So see, the pattern is very clear here. If there's no alternative splicing, you would expect the joining would be from the axon to the next. But when you have alternative splicing, you see such patterns. Okay, so this is called sashimi plot, and it's a great tool to study alternative splicing. And it's very easy to generate from IGV. Just right click, and then you have this sashimi plot. Okay. And if you can zoom in, let me zoom in. So you actually can also see the mutations. Okay, let me see if there's any mutation. Okay. Well, in this case, it doesn't seem that there is any mutation. Oh, right here. Okay, so, and if you zoom in, well, this is probably not a good example. So see, these are indicating there is a mutation here. Okay, but the confidence level is very low because there's only one read indicating this is a variant. But of course, this is not a real. Oh no, hold on. I was wrong. So in the reference genome, that's a T. Oh yeah, right here. Sorry. Okay, that's very This is the how the mutation look like in the sequencing data. See. But be careful with mutation analysis in RNA sequencing. Like I said, because the cDNA reverse transcription can introduce artifacts which would appear to be variants. So in general, the calling variants from RNA sequencing is less reliable than calling this from DNA sequencing. Okay, so that's pretty much it. So again, you can write me emails if you have any questions, and all we can discuss right now. But that's all from me today.
Great, great. So does anybody have any questions for Dr. Zheng? Okay.
All right.
So do you mind if I take control of the screen again?
Yes, yes, please.
Okay. So it looks like as far as like the final project, there's really only a few people left. So I wanted to ask really quick, do you want to do a final project, or would you like to leave it where it is for this semester? So really, I think we've got Simon, Aola, Catherine, Elias, Holly, and sorry, I'm blanking on who Peralva is. I'm blanking on who that is. But yeah, if you could unmute and let me know what you want to do.
What are you thinking of putting in it? Just like everything we've done, or are you thinking of doing something just based off of this that we did today? Or
so what I was going to do was I was going to set up more virtual machines and then have you go through, find a study, get the raw data, process it, and then do the biological interpretation. So basically tying together what we've done. But to be honest with you, a lot of you have already done so much of that because we practiced that for several weeks now. The only part that's really new is getting the raw data and processing it there. And so that's where I was kind of wondering, is that something that would be worthwhile at this point? Or is there something else that would be worthwhile to do? Or what are you guys thinking?
I think I don't think it's like imperative, because like you said, we've done everything except the salmon bit we've done.
Right.
I think it'd be interesting, but I don't think it's imperative that we do it. Also, what's the time frame you had in mind? Because I wouldn't mind doing it, but it's not going to happen this week.
Yeah, that's fair. I know this is a really rough time of the semester to do a new thing. So that was my other reason for thinking about this. Okay, so I guess I'll also ask Aola and Catherine and Elias and all, yeah, and Joseph, if you guys have any thoughts on that.
Hey guys, I'm fine if we don't do the final project. I have plenty of other things to do as well.
Okay. Okay. Yeah, go ahead.
Yeah, on my end, I have a couple things that I'll be practicing from different projects. So I'll be doing like RNAseq and the ChIP-seq stuff, but I'll just be working on that, but not related to big, I guess.
Okay. So let's go ahead, and it sounds like it would be good to just end it here. And we'll give you guys back some time. And if you have any questions or you want to work on anything, I'm always available. I'll be doing my office hours. Like, that's been really convenient to have those, honestly. So I'll be doing those for the foreseeable future. So if you have any questions, you can always come ask. And hopefully I'll let you guys know soon what we're going to be doing next semester. So I've sent out a survey regarding, you know, kind of like how is the course going, you know, comments about the course, comments about how we could, you know, improve it for next time. If you get a chance, it would be really helpful if you could fill that out. I'm going to put the link in the chat here. It's really short. I know I've sent some long surveys in the past. This one's pretty short. And it would really, really help us in order to improve this and improve like the pacing and that sort of thing. So I think we found that we probably went a little bit too fast this semester. So we want to make sure that we're going at an appropriate speed and we're covering everything that's going to be useful for people. So, okay. Does anybody have any questions or comments before we break? Okay, great. So, one more thing. We're actually having, so we've got this club called BIG. It's our Bioinformatics Interest Group club. And basically it's for people who want to use these sort of techniques in their research and want to be a part of the, I guess, community of students who do that. It's for us. So if you have any interest in joining that, do, you know, feel free to reach out. We've got a meeting actually in about 13 minutes. So if you want to hang around, we're going to be doing that pretty shortly. And I'll put the link to that actually in the chat here. So if anyone's interested in going to join that, do feel free. And yeah, thank you all so much for attending this. This has honestly been a lot of fun. This is our first time doing this and we had really no idea what to expect, but I've really actually been very, very happy with how this has turned out. So thank you all for making that happen. And yeah, we'll see you next time.
Yeah, yep. See everyone next time. So Henry, thank you so much for taking the heavy lift. So it is kind of fun to watch how the student want to take the leadership, and also, I mean, those student kind of graduate getting lower. It's almost expected because it's a final time almost there. So I guess you are happy to not having the final. Maybe next time we should have a final at the very first, to sort of do the project first.
Right.
Time when you're hitting the final, I know the other course loads start getting heavier than that chance of having a little holds on everyone. It is a little difficult though. So we may have to, Henry, if you're going to run next semester, probably shorten a few weeks, shorten the whole thing a few weeks. So the
sure.
People not thinking about, okay, we have final project coming up everywhere else, and then you have to deal with too many things.
Yeah, that's a good point. Yeah, I don't know. I don't know how much you actually learn from the hands-on session, because I don't know. You know, different people have different background and so how you can coordinate. You know.
That certainly is one of the, I think it's always a difficult part for even our class as well, because hands-on, just you try to organize everyone at the same level, because somehow you have to get your head down, which is, I mean, it's difficult. I mean, it's a great that you guys all have some experience to begin with. So when you want to get into the computational side, now you have to spend more time. That only means it's probably not less than that. So it's great, Paul.
Okay, it's awesome.
All right. Well, yeah, thank you all so much. And yeah, if you want to stick around for the big meeting, do feel free to do so. But for those of you who are going to head out, yeah, we'll see you all later.
Okay. Bye-bye. Thank you, Henry. Bye-bye.
Thanks. All right.